Lecture 12.
Okay, so today we'll start the complex subject of the electrical properties of cells and discuss resting membrane potential, and then on Thursday we'll present the action potential, and that will complete the material for the second exam, which will be a week from today. I'm sure you already know, and we'll cover the lecture on articulation, which was lecture 7, which was presented actually before the Unit 1 exam, but that will be on this exam, so it will be 7 through 12, and I'll talk more in detail about the exam on Thursday. We've got the I-Clicker questions at the end of today, trying to keep it balanced, so I'll go on and get started. The material for this is from Boren and Volpape's book, chapter 6, pages 147 to 156. That's not the whole chapter. After that, he talks a lot about details about channels, which is very significant and important information. I'll pull in some of it here and there as I go through the rest of the course. But the details of that is, I think, a little beyond that. So if you'll focus on the first part of the chapter, but that in itself is fairly detailed and difficult reading. So I think what you should do with only a week before the lecture, if you're not familiar with the resting membrane potential and its generation, really learn it at the level I presented in the lecture. And you can go to some other more basic physiology books. Vernon Levy has one called Principles of Physiology, but find some book if you're having difficulty with Boren and Volpape on this information. That a general physiology book may help. Okay, so the fact that cell membranes demonstrate electrical properties gives us physiological capacity in many different areas. For example, as listed on the outline, what we usually think of first is the ability of cells to communicate with each other, i.e. the whole nervous system is dependent on a signal being generated in one part of our system or one organ and going to another organ or cell for changes in activity. So it allows cells to communicate with another cell. Muscle contraction through that whole excitation, contraction, coupling process we've covered already is part of this electrical properties of cells. Many different hormones, although not all, are controlled by electrical signaling, changes in membrane potential, and so forth, as we'll discuss second semester in more detail. And finally, even the constituency of cells, the ion concentration gradients, are to a measure determined by the electrical properties of the cells we'll talk about today. Our earliest understanding of the fact that electricity, quote unquote, was involved in biological organisms was studied in the late 1700s, about 1790, by Galvani, when they were first finding out what electricity was about. And these experiments were shown on one of the pages of this chapter, and it just struck me so interesting that I wanted to just mention a couple things. So what he did, for example, was he dissected a frog and put it in a bell jar, grounded it to the water in the well, and then coupled it through a wire up through a couple of insulators, glass tubing essentially, up to a lightning rod. So he called it natural electricity, which is what they called lightning at that time. So he found out the effect of lightning on the dissected frog, and which looks like this, by the way. So every muscle in the frog contracts, the tongue extrudes, and essentially every muscle in the frog goes into tetany. I have a couple problems. How long does the frog have to be dissected and sitting out there while you're waiting for the lightning? And where do you find a graduate student willing to stand there and look at the frog in the bell jar waiting for lightning to strike when the lightning's going to be striking about 10 feet from where you're standing? And how many of the graduate students ended up like the frog? Anyway, kind of funny to think about how they did these original experiments. But we have made a little bit of progress since that time, and now we can record intracellular voltage gradients just by use of electrodes. This was first done by Huxley with the SWID axon, which is the largest neuron we know of. And we first started learning about what the amplitude of these potential differences are and what action potentials are from these experiments with the SQUID. But now we can record potentials in the smallest of neurons by means of microelectrodes and see how those potentials are affected by various gradients in ions and electrodes and stimulations. So, for example, we can put a microelectrode in a cell body of a neuron and stimulate that neuron by a second electrode or other means and record the changes in potential that occur in response to stimulation. And more recently, we've even got fluorescent dyes that alter the amplitude of their emission in response to changes in membrane potentials. So, as shown here, the change in fluorescence exactly parallels the change in potential that you can record with electrode. This gave us the added advantage of not studying potential differences in the cell that had been penetrated by electrode at all. But both of those give fairly consistent responses that can be recorded. From that, we learned that all cells have a very small potential difference that occurs across them. Not only neurons, but all cells have a small potential difference. And we call that the resting potential. And the genesis of that occurs because of diffusion. So from studies by Nernst and coworkers, we developed the term diffusion potential. You have already been presented in this class, and probably in many others, the fact that active transport mechanisms, most importantly sodium potassium ATPase, creates a very large difference in concentration of some charged elements, mostly sodium and potassium. So we call these ions, and the concentration difference is a significant concentration difference of these ions across membranes, largely because of active transport. We've also talked about the fact already in this class that membranes differ in their solubility and permeability of different substances. And ions are a great example, where some ions can diffuse through channels quite readily, others are almost totally impermeable. So these two properties are very significant in the development of a potential difference. In other words, we first form differences in concentration, and then some of those ions are more permeable than others and diffuse down these gradients and establish a potential difference. And let me explain this quite simply. This is that sodium-potassium ATPase we've talked about that's part of every biological membrane. And as I've said before, we extrude 3 sodiums in exchange for an influx of 2 potassiums. So charge is shuttled across that membrane by this transmembrane protein, or integral protein of all biological membranes. The little wiggly line in the middle of my little circle, as an engineer will tell you, we've got several of the class, denotes in schematic drawings a motor, some active element, and that responsible for the energy on this transporter, as you know, is hydrolysis of ATP. So every time one ATP molecule is hydrolyzed, 3 sodiums are expelled in exchange for 2 potassiums. And the result of that is what's given here as large gradients in ions. So you end up with much more sodium outside of cells than inside. We keep sodium low. And the opposite orientation for the other cation, potassium. So potassium is elevated inside in comparison to potassium outside. Then the next step in the formation of a diffusion potential would be a difference in solubility or permeability through the membrane, and indeed potassium can diffuse out of the cell and does so. So potassium goes down its concentration gradient diffusing out of the cell. Sodium, in contrast, can't diffuse in. It's about 200 times less permeable than is potassium. So much more potassium has ability to diffuse out than sodium to diffuse in. So we start forming a difference in cation concentration. And as potassium diffuses out, you're essentially moving positive ions or positive charges out of the cell. So we can start creating a difference in concentration. So sodium being pumped out, potassium pumped in. The high potassium is diffusing, which is a passive process, out of the cell down its concentration gradient and creating a potential gradient. So I said there's a net sodium or potassium efflux due to a chemical concentration gradient, which starts establishing a potential gradient. Now, the next step in the process is this elevated concentration of charge or increasing amount of charge will start a second unidirectional flux, and this one is an inward flux of potassium going back into the cell down its potential gradient. So as potassium starts forming a concentrated caused, if you will, potential gradient, potassium starts moving in down its potential gradient. And so you have two directions of flux or two unidirectional fluxes that continue until an equilibrium is occurred. And that equilibrium occurs when the efflux due to a concentration gradient is equal to the influx due to potential gradient. And that's what we call electrochemical gradient. So we have the same amount of ions moving each way across that cell, and so we're in equilibrium, and there's potassium ions moving in down the potential gradient and out down the concentration gradient, but eventually that will occur and reach an equilibrium. So we have chemical versus potential equilibrium, so we call electrochemical equilibrium. A man named Nernst figured out experimentally, he actually had to experiment to find this number. It's not anything you could reason biologically. But he discovered that this electrochemical gradient was equal to this relationship described as what we now call the Nernst equation, and that is the membrane potential that occurs at this electrochemical gradient is equal to minus RT over ZF times the natural log of the potassium gradient, the potassium inside the cell over the potassium outside the cell. And there could be conceivably a Nernst equation, as you will see in a minute, for any ion, but right now we're just looking at the most permeant ion in most biological systems, which is potassium. The R in this equation is the R which we always use, the symbol which we always use for the gas constant. It's 0.082 liter atmospheres per mole per degree Kelvin. The ion charge, Z, for potassium is 1. This calcium, of course, would be 2 and so forth. But if you have a monovalent ion, Z is 1. The Faraday constant is how much charge, how many coulombs of charge is related to a certain mass of gradient in ion. And so it's 96,500 coulombs per gram of charge. And the temperature in biological systems is usually 37 degrees centigrade, but we have to measure it in Kelvin, and that's would make this turn out to be an equation. What we usually do in biology is simplify this equation and convert all those constants to one value, which you can do, especially if you're working at a 37 degree ambient temperature. So 37 degrees body temperature, monovalent ions, which we're usually concerned with in these working with action potentials and resting potentials. And it's a lot easier for biologists to work with log the base 10 than natural logs, and so we convert this to minus 60 times the log of the potassium inside over the potassium outside. This will come out in millivolts. So this ratio tells us what potential will be generated or result from a certain difference in potassium concentration across a biological membrane. And let's give you an example of that. Usually there's about a tenfold difference in potassium. It can be less, it can be more. There's a lot of differences, and we'll explain why biological membrane. But let's say you got 10 millimoles of potassium on the outside of the cell, and as we always said, far more, let's say 100 millimoles inside. So if you have inside 100 over outside 10, then you'll get that ratio equal to 10. So the ratio of potassium outside to potassium inside concentration, 10, the log of 10 is 1, if you remember your logarithms. If not, you get out your little iPhone and calculate it. So anyway, you get a log of 1 with a tenfold difference. And so that would mean that if you had a tenfold difference of potassium, the membrane potential would be 60 millivolts, a minus 60 millivolts, which means, by the way, the inside of the cell is negative with respect to the outside. And that is a pretty common potential difference of living cells. Neurons are usually closer to minus 90, muscle minus 85, minus 80, but a lot of very active cells can be around minus 60. Epithelial cells that aren't very active have much lower membrane potentials, but all cells have a membrane potential due to this, what we call the fusion potential. To take the story one more level, I'm going to talk about reversal potential, which isn't really a difficult concept. The term sounds more difficult than what we're going to talk about. But the reversal potential is the potential in which a membrane is at, in which the given ion current, you have to define reversal potential for potassium or sodium or chloride, whatever ion you're talking about. But let's say potassium, so that would be the potential in which potassium shifts from a net outward to a net inward. So at the exact reversal potential, there's no net movement. It's an electrochemical equilibrium. The same amount of potassium is going into the cell as going out. And so reversal potential is the membrane potential of which there's no net ion current. And if it's greater, if you have a membrane potential that's greater than that reversal potential, more negative, then you'll have an ion that's moving in. So, for example, if the cell was, instead of minus 60 in the previous example, a minus 70, there'd be more potassium going in to reach equilibrium. There'd be a net inward flux of potassium. Aside here first, because you'll have trouble with this, an increase in membrane potential in biological terms means going from minus 60 to a minus 70, even though that's an absolute decrease. But we think of the term as potential difference. So when we're talking about a membrane potential, we're thinking about a potential difference. So if we go from a minus 60 to a minus 70, we've increased the potential difference across that membrane. So hence we say that the membrane potential went up, even though it's in absolute value gone down. Sorry about that. It's just, I didn't invent it. I have to carry the message. Okay, so anyway, if the membrane potential is greater than the reversal potential, or more negative, you're going to have a net inward flux of potassium, and the opposite is true if it's less. So if you have less than a voltage of minus 60 in the previous sample, potassium will rush out of the cell. So we call that the reversal potential. It's the point where you have the same flux in equal directions. And you can do the same for sodium. Here's a big leap, but sodium's reversal potential is a plus 67. It's a very high voltage. So again, if you're more higher increase, a higher voltage difference than plus 67, which means plus 40 or something. It doesn't make sense, I know. But plus 40, we call that a higher difference than you'd have a net, just like this, net inward sodium current. And that's all explained in this figure here. Okay, a little bit more detail to this whole story. So first of all, resting membrane potential is not exactly the diffusion potential. What I have got up to now doing is trying to explain what a diffusion potential is. What can cause a potential across membranes is if you had an active pump creating a concentration gradient and then an unequal ion diffusion, you can end up with a potential gradient and when that ion reaches electrochemical equilibrium. Well, you do have membrane potentials in all living cells, essentially. Some of them are extremely small, but they always exist. And again, it's a result of the two phenomena I just talked about, active transport, selective permeability. But the problem is we are not only looking at potassium, we're looking at other ions. So although potassium is by far the ion present in the highest, with the highest conductance, we call it, or permeability, chloride and sodium also contribute to the final resting membrane potential. And this is a little bit leap to see if you can follow this. If the only ion which was permeable in this membrane was potassium, and the Nernst potential was a minus 100 for potassium, then the membrane potential would be a minus 100. It'd be the same as that. But these other ions are slightly permeable, so they alter the resting membrane potential. And so the idea, and I'm going to amplify it with a little bit more discussion, but the idea is that the membrane potential is determined by the Nernst potential of all the ions that are relevant or important biologically. And what happens is their relative importance in determining the resting membrane potential is directly proportional to their conductance, or how permeable they are. So the ion that's most permeable is going to have the greatest influence on the resting membrane potential. So potassium, sodium, and chloride all contribute to the potential, but the final potential is totally dependent on how permeable or what the conductance is for a given ion. And so a group of very brilliant and original biologist, Coleman Hawkins Katz, came up with what's now called some people call it the chord equation. I still call it the GHK equation, the Coleman Hawkins Katz equation. And this is the real absolute definition of the resting membrane potential. Because what it does is it relates the concentration gradients to the different ions' permeability. So if the ion is really, really permeable, this ratio will have far more effect than if it's not. Let's say pK is 1 and pNa is 0.05. This ratio is going to have very little effect on the final potential. So the higher the permeability, the more effect a given gradient will influence the potential difference. So since the potential of potassium is much, much greater, almost 200 times greater than sodium, the membrane potential is usually very close to potassium diffusion potential. If you're quick and following this, which have to be fairly sharp to get the first time, but if you're quick and been following it, you realize I flipped the numerator and denominator and at the same time changed the potential on or the charge on the gradient. In other words, I changed this from a minus to a plus, and you can do that if you invert the numerator and denominator on a log relationship. It doesn't make any difference. In other words, you'll see this equation written both ways with the negative in front and all the ions that are positive inside the cell on top of the equation and outside on the bottom equation. And another thing you'll see if you look at it for a few minutes is chloride is the opposite orientation, inside or outside in the other, and that's just because it's a negative ion. It's going to go the opposite way that the others do. It's an anion. So the anion contributors have to be reversed as far as their contribution. Once again, you could almost describe exactly what goes on in the resting membrane potential by looking at potassium. Because there is, however, differences in the relative permeability of these contributing ions, resting membrane potentials vary. And so it is important. I'm not trying to say potassium is the whole story, but some cells are far more importantly permeable to sodium, and some are impermeable to all of the ions. So anyway, there's a lot of variability because of the difference in permeability. So as I said before, neural resting membrane potentials are about a minus 90 millivolts. That's quite high polarity, the biological membrane. Muscles, minus 50 to minus 70, epithelia, just a few millivolts. So all of those things will affect the cell, even cells that don't conduct information like neurons. For example, we have to have high resting membrane potentials in order to control muscle activity. So even though these are just a few millivolts in epithelia, that does affect the cell's function, as we'll see. So again, what affects these gradients or differences in resting membrane is these relative permeabilities of the different specific ions. Let me move that. Yeah. This turns out to be a clinically astoundingly important thing, and I'm going to have you think about this in my ending statement today and how it affects us. But our extracellular potassium is about 5 millimoles. We have very, very low extracellular potassium. And so any changes in that, even the type of food we eat or things that alter kidney function so that the potassium balance is affected, will markedly affect our resting membrane potential. And so on the bottom, so we have potassium concentration in millimoles. Like I said, we're normally somewhere 2.5 to 5. And the membrane potential is just radically affected by this. So small changes in potassium will radically affect our resting membrane potential. In fact, the electrolyte disorder that affect people more and cause emergency room visits is potassium. More people report to emergency rooms with potassium disparities or abnormal potassium concentrations than any other ion. It's very important biologically. Another great story. So, sodium potassium ATPase is electrogenic, and this is the first time I get to talk about the significance of that. But we don't pump these cations one-to-one across the membrane. There's 3 sodiums coming out, there's 2 potassium. So by itself, the pump creates a potential. I've been doing this whole thing about what the resting membrane potentials do to the fusion, but we're unequally translocating cations, and we're pumping more sodium out than potassium in. So that is what? That's increasing. I said the resting membrane potential is about minus 60. That's increasing the amplitude of potential. So by itself, the pump is electrogenic, creates charge difference because of the inequity of cation separation. And so there's a net efflux of cations, which makes the outside of the membrane positive with respect to the inside. So that would amplify the diffusion potential that's created usually by potassium. How important is that? It took us a long time to figure this out. Is it responsible for 50% of the normal potential at rest? Is it responsible for 20%? How important is this electrogenicity of the sodium potassium ATPase as far as the normal resting membrane potential? Well, it does depend on the membrane, but most of the time it's 2 to 5 millivolts. So it's amazingly little. In other words, it's true that the pump is electrogenic, but the the actual amplitude of charge created by the pump is almost unimportant. It's negligible as far as normal functions concerned. Okay, this is not in this Gibbs-Donan equilibrium story is not in the same chapter, so I flipped page 133 and 134. It's given in another chapter in the book, and it's not easy, but it's also not as near as important as what I've been talking about. So definitely make sure you understand the rest of the story, and then in your tons of extra time you have in your life, look at Gibbs-Donan. So Gibbs-Donan is the effect of non-permeable charged ions on this story. So if a given solute is not permeable, how would it affect the resting membrane potential? And cytoplasm contains not only charged solute, which we call ions, but also some solute that is charged but not permeant. So we not only have solute that's charged and non-charged, but we have both charged and non-charged, permeant and non-permeant solute. So does the fact that you have locked-in ions or charged solute affect the resting membrane potential? And yes, it does. It's not a huge effect, but it definitely does. So the Gibbs-Donan describes steady-state properties of a mixture of non-permeable and permeable charged solute. And it's going to be important in a couple interesting ways, although it turns out not as important as you might presume. And the easiest way I've ever tried to explain this to students is with, like every book does, with a model. And so let's imagine you start out with a solution across a semi-permeable membrane that's something like a biological membrane, and you have a solution in compartment A and a solution in compartment B. And you start out with them having the same number of cations and anions. And so over here you have potassium chloride, 0.1 molar, but over here you have potassium with another anion. And that anion is not permeable. So we could call this protein, but it would be difficult to have 0.1 molar potassium and 0.1 molar protein. But for the sake of discussion, let's say we have a negatively charged protein, which most of them are, and can't go across, and then you have potassium equal on both sides and chloride. So that's the initial conditions. And always membranes are quite permeable to potassium chloride compared to whatever this negatively charged Y is. And finally, across biological membranes, the compartments are usually isosmotic because almost all are extremely water permeable. And also, you always have electron neutrality because charges will balance across membranes. So given the initial condition, what would you expect to happen towards equilibrium? Well, there's no chloride in A, so chloride is permeable. It's not as permeable as potassium, but it's definitely trying to diffuse down its concentration gradient. So you'll eventually have chloride diffusing down its concentration gradient, and as it does so, negative or anions are moving into A, and so you're going to have potassiums move out in order to balance it. So it's going to turn out at equilibrium, like you always are going to have. You're going to have a charge equilibrium, or electron neutrality we call it. So you have the same amount of positive and negatively charged on this side, and also positive and negative charges on this side, although on the A side, some of the anions will be made up by the impermeable Y. So that's the final situation. You won't have potassium chloride being equal on this side because you have negative charges over here. You do have equality in B. So anyway, what happens in such a sequence is you're going to have electrochemical equilibrium of the permeant ions, potassium and chloride, and isosmolar, or you'll have more solute on the left than your right, and that leads me to the next story. So what happens is this starts creating a charge by itself. In other words, the fact that you have trapped negative ions on one side starts creating a charge. There's more, although you have an equal number of positive and negative charges on each side, there's a disparity of charges on A versus B because of the trapped ions. And usually this will create a potential difference by itself. And again, it's not much more. It's about the same usually, or a little bit more than the potassium ATPase will cause. But it will cause a cell potential by itself if it gives donut effect. But what also it does is interesting. It creates an osmotic pressure. It's going to should solute through the charge separation that we talked about. And so what will eventually happen is water will diffuse from B to A because you end up with more solute over here. So Gibbs donut does cause effects. They're not major effects, but it causes shifts in water because it shifts solute, and it causes small potential differences. And those are important across capillaries. We'll talk about it, and water shifts in between cells. We'll talk about it later. I just mentioned a couple of physiological applications. Looks like I knocked the thing off again. But the applications are that proteins are negatively charged at biological pHs. They're confined within cells. There's very little extracellular protein, and within some confined compartments like the vasculature. And the result is that will shift non-confined ions of water across membranes. And usually it's not more, it's 5 to 10% shift, but it does affect compartment size and resting membrane potential. Usually it's an elevation of membrane potential. But we're going to talk about that and when it's significant other times in the course. So anyway, that's another whole story because of the fact of charged impermeant ions. Okay, well, I've covered a lot of material in a short time. Definitely it's a very important and difficult subject. I would suggest, really understanding the lecture and then reading about it and perhaps some other fairly simple stories. But it's a fairly complex but important story. And we'll go from here next time and talk about how we use resting membrane potential as a source of all kinds of things like action potentials. Okay, got some eye clicker questions. A couple of these are from the past lecture, but a couple of them also will help me explain some other ideas in today's lecture. So let's see how well you're understanding the lecture also with these eye clicker questions. Okay, so this is from last week, obviously. Which of these muscles is the vastus lateralis? This is what you're doing in lab this week, so I'm a little ahead of lab. Oh, thank you. Oops, sorry. Oh, now I stopped it again. No, it's going. Having a hard time with all the buttons. Okay, what is the vastus lateralis muscle? These are the quadriceps group, the anterior thigh, obviously. Looks like we're almost all checked in, 143 or so usually. That's 144. A couple more seconds, go to 50. Okay, that'll be enough. Okay, let's see what you did. Everybody probably has this. 97%. Let's see which one. You had 97% C, so that's right. It's this muscle right here. So it's on the lateral side, and it's part of the quadriceps, the lateralis, and the rectus femoris, and the rectus whatever. So the quadriceps group. Okay, this muscle functions to what? I'll start it. Did it start? Yeah. Okay, about 8 more seconds. 5 seconds, 4, 3, last couple people check in. There you go. Okay. You can tell by where its insertion is. It's inserted on the lower leg of the anterior part, so it's a leg extender, and you all have that, so E is the right answer. Okay, number 3. Now I got some fun questions. So see if you can reason based on what we talked about today. I'll give you some time to think about this. So what would be the resting membrane potential if the conductance of potassium and chloride, G stands for conductance, so if those two aren't permeable at all, what would be the resting membrane potential of this cell? That's a typical question. Talk to the smart guy to your right. Okay, let's, I'll go to about 50 or so. I want to go ahead and check in. Okay, about 5 seconds. Go ahead. 4. Got a couple more people need to log in. Okay, that's it. See how many followed the story. Whoops, whoops, whoops. Stop. That's not what I want. This is what I want. Some people did that. How do I go back a question? Can I go back and what? Go back on the slide one? Graph. I don't. Oh, back. Oh, there you go. Thank you. You guys, how come you know so much about this thing and I don't? Oh, there it is. Thank you. Thank you. Okay, so most 76% of you selected A. Let's look over at what A means. That'd be a very hard time. Okay, there's A, and now I need to slide this over. Okay, so A is the membrane voltage if the membrane was essentially a electrochemical equilibrium for sodium, and that would be 65 millivolts, which is true, because it's the most conductive ion. So if the other two can't conduct at all, even though sodium conduction is so slow, sodium will eventually reach electrochemical gradient at plus 65. And that's an important, really important concept for where we're going on Thursday. So definitely 75% of you are following the story, which is really good. If chloride wasn't permeable and sodium wasn't permeable, and the membrane potential would be exactly the potassium, or a minus 100. If chloride was the most permeable ion, then it would be a minus 90 and so forth. We have some, often the question on exams is asked like this. We have some chemicals called ionophores, and they're essentially artificially inserted channels that make a membrane selective for a given agent. So I could inject into a given in vitro system an ionophore for chloride, and that will make chloride tremendously permeable. And so then the membrane would be at a minus 90. So in other words, there's lots of ways that I could ask this question. If I block this channel, if I made this channel most important, what happens to the membrane potential? So anyway, that should be a really easy concept. So I introduce it right here at the beginning so you can follow it. Now the second one is just as important but interesting, and that is, what would be the effect if you added a biologically effective dose of Wabane to the resting membrane potential? So you have a minus, let's say you have a minus 90 millivolt potential, and I added Wabane, what would happen? Okay, about 10 seconds. Okay, 2 more people, 1, 2. Okay. See if I can hit the right buttons here. Okay, the majority of you aren't always right, but this time you are. The cell will not immediately depolarize, and why is that? Well, you've got the resting membrane potential already established. Now you stop the machine that generates the gradient, and the gradient's still there. So what has to happen is slowly the ions have to equilibrate in concentration, and that takes a long time. The sodium, for example, just is not very permeable at all. So for sodium to leak down its concentration gradient so the cell will depolarize, in some membranes will take hours. It depends actually on the permeability gradients and final conductances, but usually with 10 to the minus 4 molar Wabane, it will be hours to get back to 0. So you have a neuron, you've got an electrode in it, and find its resting memory potential is minus 60, you add some Wabane, and you watch it go up and up and up, and it'll take for a long time. So what has to happen is potassium can go out pretty readily. It'll equalize pretty readily, but sodium, it takes forever and ever for it to equilibrate. So it will take a long time to reach a 0 potential. So that's kind of an interesting realization, and it helps you know a couple things. One is that if you poison some potassium ATPase, you can have neuron activity for a long period of time because there's this large store of ions. And so we'll talk about that next time when we talk about action potentials and how much shift occurs in given situations. So slow sodium influx, it takes a tremendous amount of time to depolarize the membrane. Okay, so the last question. Okay. I guess you have the capacity to change your mind. Can you erase? I see some people chose Spartan and then it disappeared. I was hoping more people chose A. Okay, a couple more seconds. And that's 1:43, 44. Maybe they snuck out the back door. 2 seconds, 1 second. There's 2 people. Okay, they're there. Trying to get y'all in here. Okay, so now we got the stairs, the Truppy effect there, with the majority choosing D, but there's some nice votes on the other side. So I was hoping more people would choose A or E, which could be true, definitely. Probably were, actually, when I drew this. But the real answer is, just to show you the relative effect of Wabane, and that is that it will still, you'll still have, when Wabane kills it, you'll depolarize the cell, but only a couple millivolts. So that effect is minor. And that is the right direction, of course. You would have less polarity with Wabane, and then we'll slowly go up the rest of the way. And that effect will occur immediately. So there's a couple pieces of information. The sodium potassium electrogenic effect is immediate, but the diffusion effect is long-term. Okay, a lot of information to fool with, and I'll see you on Thursday.